It's a great pleasure to be here and have the chance to talk to you about the type of research that I'm really burning for, network and network biology. So network biology is a pretty big field. In the 25 to 30 minutes we have available, I'm not going to try to cover that. We'd all leave the room with head spinning and scream for lunch. Instead, I'll try to narrow it down and tell a very streamlined story, starting from large scale, talking about networks among humans, and take it down to the molecular level and biochemical networks. And hopefully, at the end of this talk, you'll see, okay, now we understand why he's involved in this stuff. So, quite short introduction to what my group is doing. We have 3 directions of research. Network analysis, and that could be applied to a wide variety of problems, not just biological. We do a lot of metabolic network modeling and also stochastic analysis of genetic circuits. Here's a little example of that. All of the work is computational and mathematical, analytical. And I try to stay honest by keeping out of the lab. I discovered early on that's a safe place for me to not be. So today we'll start out with some of the network analysis parts and slide over into the metabolic modeling. So to introduce and get a common ground for what do I mean when I say network, I put up this example. So if you look at this, I hope that some of you can recognize what it is. Anybody want to dare to raise a hand just to show that you understand this? I'm not going to call on you. All right. So if you look at the names, you might recognize some of them. Frodo, Elrond, etc., etc. The social network of Lord of the Rings. So this was put together by some students who slaved for a few weeks. Well, maybe not slaved. They pretty much enjoyed rereading Lord of the Rings. And every time 2 characters talk to each other, you put a little notch for that link. So the thickness of the links represents how many times they interact through the book. And by not knowing anything about the plot of the book, you can look at this network and start analyzing the contents and the structure. Say, who is the most important character in the book, meaning most central when it comes to who interacts with whom, who are most closely related, etc., etc. And you'll end up with the cast of usual suspects. I'm not going to spoil the story for you if you haven't read it, but it's worthwhile check into. So right now we'll go into the part of large-scale network structure. And in order to talk about that, a good example is spreading of traits. It could be spreading of disease, but I think of traits as a bit more closely related to what we're looking at the hunt type of studies. Smoking. Is that a trait that is spreading in a network? Well, I asked the question and you can pretty much guess what the answer should be. And yes, it is something that spreads. But if you just sit down and think about it, you say, no, that's my choice to not smoke, and that's those people over there's choice to smoke, and I will stay away from them so I don't get infected with that lust for smoking. So this is a view from a Framingham study, 1971, where people were asked lots of questions about what they were doing, how they were feeling about themselves, their weight, if they were smoking, their income, who were their friends. So they gave a list of top 10 friends. That doesn't mean that if I put Christian on my top 10 list, that he is going to put me on his list. You know, we'll have to see about that. But so these links, they are one way. If both people report a mutual relationship, it is a two-way link. And you'd think that that's a much stronger link. So this is a snapshot from 1971. And this is from year 2000. People who smoke are in yellowish, and the number of cigarettes per day is the size of the circles. So if you look at the difference between these two networks, you see that the people who smoke, they are not as central in the network as before. And they're sort of pushed out on the outside of the network, if you can call it that. So if you do some statistical analysis, you can tease out what is actually spreading on a network when you adjust for different confounding factors. And is the stopping of smoking, if your friend, what you perceive as a friend, stops smoking, there's a decrease in the risk of you smoking. That's about, if I can read this sideways, 40%. Now, if your spouse stops smoking, that has a significant impact on your chance of stopping to smoke. Great. So what about other traits? Happiness. Is that something that spreads in the network? Isn't that something that's just you deciding if you're going to be happy, or you pop some of the happy pills? Well, it is a network trait too. And as we go through another example after this, you'll see that many traits spread in network, and statistical significance is measured out to 3 steps in a network. So people you don't list up as your best friends will affect the chance that you will have a certain trait, increase it or reduce it. So what about happiness? 1996 and 2000 network, we see that there are some differences in the structure. When you do the number crunching on the network, look at friends and friends of friends, and look at the reported levels of happiness, you can look at these results. So distance is a very important factor in happiness spreading. So if you're closely related to neighbors that are not far away, friends that are not far away, will have much higher impact, those that are physically further away from you. So nearby friends, they can have an increase in probability of happiness. If they report happy, they will increase your chance of reporting happiness. Distant friends, much less. One thing that I found really strange is co-resident spouse has almost no impact. I leave that without a comment, and we just quickly go on to the next issue. Network of human diseases. My claim is that this demonstrates non-intuitive structure and close connection with biochemical and genetic networks. So that's my claim, and try to keep that in mind as we go through the next few slides. So you can start building up a network of diseases by looking at, for instance, disease phenome, that is different diseases, and those that have, and the genes that are known to be related to the diseases. A good source for this is the OMIM database. There are other ways of approaching it, but this is one particular way that's received quite a lot of attention. So this is what's so often called the bipartite network. You have 2 kinds of nodes. You have diseases and you have genes. And diseases aren't directly connected to each other. They're only connected to the genes that are associated with that disease. However, it's possible to make 2 separate networks out of this. If you say, I look at this disease and all of its neighboring genes, and I hop over to other diseases that they are connected to. So I sort of trace out and get rid of all the genes. And this is the human disease network. You put in links between the 2 diseases if they share a genetic cause or association. The disease gene network is links between 2 genes if they're involved in the same disease. So what would you expect this resulting network to be? If science was really simple and we wouldn't have a job, then you'd have only 1 disease and 1 gene. You'd end up with singletons, as it's called in this network. Not exactly what we see, is it? So this is a picture of the giant component in the network, meaning of the different diseases that were studied, about 2/3 are in this connected system where you can start at one disease and follow a link to a different disease and follow a link to a different disease and follow a link, etc., and you can go around this whole loop in this system. So you would expect that diseases that are neighbors should be closely related. And of course, that's also what you see. The different colors are related to the disease classifications. But you see, you can also go from one disease to something that is totally different by just following these links of shared origin. And I hope that now you start to think, well, wouldn't it be nice to know why that gene is associated with those multiple diseases? How can we connect them? Is it just some, is this just caused by the crude level of resolution, or is there something more? Some properties of the human disease network. The giant component is, as I said, about 2/3 of the network. Most diseases only relate to a few genes, and a few diseases consist of dozens of genes, and these are the biggest ones. And the most connected disease classes are cancers and neurological diseases. And in the same disorder class, you can say, if I just randomized my system and then say, what would that network look like? Breaking the correlations between diseases, in a sense. You should have about 107 links within an average disorder class. And instead, you see much larger numbers. So there's a signal in this. So you might think that just looking at diseases, well, it's a cute little network thing. But does it really matter? Yes, it does. Because it's related to disease comorbidity. So this is a slightly different representation. Here's a metabolic network, different enzymes, and the names of the genes coding for the enzymes. And then you have the different diseases and genes associated with these diseases. So you can think of it as two different networks, a biochemical network and this sort of artificial disease network. And you can smash it together and start looking at what are the traits that we see. Can we relate this, what we see of interactions in the disease network, to connections in the biochemical network underlying the activity in each cell? And we do find statistically significant correlations between cellular interactions and comorbidity pattern. Going through Medicare data, we're talking about many millions of Medicare data, looking at the IDC codes and seeing what type of diseases happen. What is the chance of getting a disease if you're coded with one particular disease? And you see that neighboring diseases in the network have a higher chance of co-occurring than if they're far away in the network. So there is a propagation, sort of spreading effect also in this network for disease development. So this is a large-scale view of that metabolic disease network. The different nodes are the links, are the diseases, and links correspond to interactions in the biochemical networks. The size of the links correspond to the strength of the comorbidity score. So we see that there is quite a big level of heterogeneity. Not all the links are of equal importance. Some are highly significant, and the red ones are those that are statistically significant in the system, and others are not so significant. So at this point, I hope that you see there is a connection between biochemical networks and the higher level disease systems that we're used to think about in humans. Now let's switch gears a bit to constraint-based analysis. The reason for this is that there are many tools now that you can use in order to study biochemical networks. We started up there with human interaction networks down to biochemical networks. And it's a very solid mathematical foundation for the predictive modeling of biochemical networks. And nowadays you have lots of tools available, making it easy to access. If you have some good biochemistry background, it means that it's very possible for you to make good interpretations of the data you get. So this is a very short crash course of the constraint-based analysis. Consider a cell a bag of chemicals. Look at each chemical. Which ways can it be produced and which ways can it be consumed? And you set up a mass conservation and steady state constraint for that metabolite. You do that for all of the metabolites. And you can formulate the system as a set of linear equations. Pretty easy, huh? We could do this in junior high school. The only challenge is that this set of linear equations is underdetermined. So what do we do now? We can use optimizations. We can apply more constraints. If you have information about your system, you can bake it into the formulation through constraints. So if you use this standard method, that's called flux balance analysis, and it performs really well on bacteria. And you can predict gene knockout strains in E. coli and yeast with about 90% accuracy. How are you able to do that? You have a good genome annotation. And then you build a model, you test it against different growth experiments, and then you get that high level of accuracy in predicting phenotypes. This is using the same model on yeast. And whoops, we see that something is not going so well. This is among the best models available. These are experimental data points for yeast growth. This is growth rates, and this is glucose uptake. So as the glucose uptake rate increases, growth increases up to about a level of 0.3, and then it plateaus. This computational modeling just says it should go forever up, 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 up. More glucose, more growth, excellent. But that's not what happens. How can you understand this? The cell has internal limitations. It's like a zero-sum game. A cell has only a certain amount of volume available, or you can say the proteome must be a certain mass fraction of a cell. That mass fraction can change if the cell is growing slowly or fast, but it is a limit. You can't just say, I'm going to have many more of these proteins because with those present, I can grow much faster. You need these and those and others, and all of a sudden you have a need for more proteins than you can fit into a cell. So that means it's a zero-sum game. You have to give and take and figure out what is the best allocation of your resources to get growth. So we can introduce that into this modeling, and then we get these curves. If you calculate correlations, you get correlations for this horrible fit. That's about 0.98. So if you don't look at the curves, you get a correlation of 0.98 and say, yes, I'm going home. But if you're a good scientist, you should not do that, of course. So in this model formulation, we needed turnover numbers for different enzymes in order to introduce and include this resource restriction. What if these turnover numbers aren't really good, maybe not totally trustworthy? Because they're fetched from many different sources, not one consistent type of experiment to figure them out. So we have devised a method to say, well, how can we adjust these turnover numbers to see if it's possible to fit the data? Is it at all possible? So we use this method, and we see that we only need 5 turnover numbers to change the value. This is the scale of change. So this one, TDH1, its turnover number needs to be 5 times as much as the original number from the databases. And the others also need to be scaled up a little bit. And this plot shows that as we use our method with more and more iterations, the result is really robust and stable. We get the same result no matter how many iterations we use if we go past about 200-300 iterations. So it's a very solid result. Now what is the consequence for the phenotype of the cell? Change those 5 and we get these curves. So for the growth curve, we see the saturation happening. And for the oxygen uptake, which we did not use in the fitting anywhere, we get a wonderful match. Now what do we take into this modeling paradigm? We just use a list of biochemical reactions, and turnover numbers, and then some mathematical principles. We don't say anything about toxicity, don't say anything about regulation, or other aspects that are really, really important. But this is a powerful method because it goes to the core of what is necessary for a cell to function, metabolism. We also tested what if we just randomize the turnover numbers. Say, we don't trust any of them, but we want to say we put turnover numbers that follow the same distribution that you get from the data set. And you see that it falls into these two different categories that correspond to two different ways of using the metabolic network to about the same growth rates. One is having a high carbon import, and the other has a higher oxygen import. So two different sets of pathways are activated in order for these cells to perform. And we can also look at these correlations between fluxes in the system and relate that to the different turnover numbers that we use. And we see that there is a certain pattern of behavior that the network enforces by itself. So this plot shows that something called the Jacquard Index, which is a measure of similarity between two sets. One, that is the reactions that should be tightly connected just based upon the biochemical network structure. And the other based on correlations measured in these computational fluxes. For many of the reactions, they have the same sets. Not really interesting. It means that these sets are purely determined by structure of the network. However, we also find some sets that are highly correlated that are not driven by the biochemistry of the system, the obvious biochemistry of the system. So to summarize, my claim is that biological interaction networks directly affect connections between diseases. This constraint-based genome-scale metabolic modeling paradigm is one way of bridging biochemistry and up to large-scale predictions of how a system behaves. And since it's constraint-based, it is not very taxing to add new information to your system. You formulate it as constraints. As long as you can do that, you're in business. And it is also possible to combine this in different ways with other types of interaction networks that I haven't talked about today. I only had 30 minutes. And that's protein interaction network, gene expression correlation networks, etc., which also give different views into the molecular level of activity in an organism. So with that, I'll say thank you very much. And thanks to all the people who have participated in this work and different aspects of the work.